முட்டை மட்டும் தனியா சாப்பிடுவேன். ஆம்லேட் தானே, ஆப்பாயில் இல்ல. ஆப்பாயில் சொல்ல போனா முட்டை எல்லாம் தனியா சாப்பிட்டா அது ஒரு டேஸ்டா. வச்சுட்டு வாடி போயிட்டு வரும். பிச்சு விட்டேன் என்ன கூப்பிட்டாங்கன்னு என்ன கூறியா நினைக்கிறது.
Good afternoon. This is our biochemistry lecture for this afternoon. We will have two: catabolism of proteins and good afternoon. This is our biochemistry lecture for this afternoon. We will have two: metabolism of proteins and Okay, sorry for that. Okay, so we will have catabolism of proteins and of amino acid nitrogens and carbon skeletons. Okay. For those who won't fit in the Zoom account, I have shared with your LO the YouTube link so that you can join our live stream for this lecture. And you can go back to the lecture if you're not able to, if you want to review for your review for the exam. Okay. The objectives for this lecture is to explain the metabolic fate of amino acids that are catabolized in the body. Okay, you have learned previously what are amino acids, and now we will learn how they are catabolized and how our body utilizes these amino acids. Okay, and we identify the major enzyme systems which would be responsible for degrading damaged or proteins that we don't need anymore, such as your ubiquitin, transaminases, and your glutamate dehydrogenase. Okay. Some objectives would be knowing the role of the stomach, the pancreas, the small intestine in protein degradation. Okay. We look at the role of the urea cycle as it metabolizes our nitrogen. Identify sources of nitrogen atoms in the urea and know the symptoms for your clinical, know the symptoms of hyperammonemia and their causes. Okay. The biomedical importance of your protein and amino acid catabolism largely hinges on how nitrogen of your amino acids would be converted to urea. Okay. You will know that your nitrogen, if accumulated, would produce certain symptoms that would be toxic to our body. Okay. Because in equilibrium, nitrogen should equal intake equal output. If there's deficit, positive or negative nitrogen imbalance, then disease will occur. Okay. So if there's positive nitrogen balance, so there's an excess of ingested nitrogen. So this is usually evident in pregnancy, if there are growth spurts, and the like. If it's negative, where output exceeds intake, this is usually followed by surgery, cancer situations, kwashiorkor, and marasmus, your nutritional deficiencies. Okay. Clear so far? Okay. So this ammonia, which is primarily derived from your alpha amino nitrogen of amino acids, is toxic, as I have mentioned, and is converted to ammonia to the amide nitrogen of non-toxic glutamine. Okay. So this deamination of glutamine would release your ammonia, which is then converted to your non-toxic urea. However, if your liver functions compromised, for example, as in cirrhosis or hepatitis, ammonia will elevate and, of course, would produce some clinical signs and symptoms. Okay. So we ingest your protein. What are our usual sources of protein? It could be animal fat, animal protein from chicken, feet, chicken, meat, liver, etc., etc. It could also be plant protein, for example, your tofu, etc., soy, and it will be digested in your stomach and then converted into your amino acids. Okay. And these amino acids would then be metabolized and will be used in the liver. Okay. So these dietary proteins will go through our small intestine, and it will be it will be reacted upon on by different enzymes, and thereby produce your different amino acids along your chain. Okay. So usually our dietary protein, we ingest around 100 grams per day. Okay. What will happen? Of course, it will turn into your glucose, your ketone bodies would be into your body protein. And what's important here in your catabolism is by your urea would be excreted by your kidney up to 30 grams per day. So our body has a limit on the protein that can be catabolized, okay, such that any excess, of course, in our diet could produce disease. Okay. So protein turnover occurs in all forms of life. Okay. Our total protein would be 1 to 2% turnover. Primarily, this is muscle protein. Okay. If there's high rates of protein degradation, okay, that will undergo structural rearrangement. Okay, for example, tissue growth such as in pregnancy, or any changes, for example, in lower life forms, your tadpole metamorphosis into your frog, okay, or skeletal muscle when you do starvation. So there's a lot of protein deprivation or protein excess, then there would be a lot of degradation occurring. Okay. Of the amino acids, we can reuse up to 75% of amino acids. That's how conserving our body would be. But the excess nitrogen will form urea. Okay, you cannot store these amino acids, and those not immediately incorporated into new protein are rapidly degraded. Okay, so how are they degraded? So these are through proteases and peptidases. Okay, the susceptibility of a protein to degradation is expressed, as you know, as its half-life. And the half-lives of liver proteins would range from under 30 minutes to over 150. Some housekeeping enzymes would have 100 hours as their half-life. And some key regulatory enzymes have a shorter half-life of 0.5 to 2 hours. Okay. So there are pest sequences. So your pest would be composed of your proline, your glutamate, serine, and threonine. And they target some proteins for rapid degradation. Okay. And this resulting peptides would then further be degraded to amino acids by your endopeptidases that cleave internal bonds. Okay. Your aminopeptidases and your carboxypeptidases remove amino acids sequentially from the amino and carboxyl terminals, respectively. Okay. They are ATP independent processes because they are extracellular, that are associated with the membrane and associated with intracellular proteins that are degraded in lysosomes, ATP independent processes. Okay. what are requiring of ATP and ubiquitin? Your other abnormal and other short-lived proteins that will occur. Of course, you know that if it occurs in your cytosol, then it requires your ATP and your ubiquitin. Okay. What is this protein that they call ubiquitin? So it's a small protein that will target your intracellular proteins for degradation. Okay. So primary structure. So it will quite resemble your yeast, human ubiquitin, and will be attached to non-peptide bonds formed between the carboxyl terminal of ubiquitin and the amino groups of lysyl residues in the target protein. Okay, so enzyme 1 would activate your enzyme. Enzyme 2 would be a ligase. And enzyme 3 would be a transferase. Okay, do you remember what is a ligase? What does a ligase do?
It joins two molecules. It has a joining function.
Two molecules together. So after activated, it joins your two molecules. And what's a transferase?
Transferase is an enzyme that transfers any molecule to the target protein. Like if it's acetyl transferase enzyme, so it will transfer an acetyl group to the protein.
So transferase. It transfers one molecule to another or just rearranges. Okay. So what does it do? The terminal of ubiquitin first forms a dioester and will be hydrolyzed by a pyrophosphate, ensuring that the reaction will proceed readily. Then this exchange gets transferred, okay, to an activated ubiquitin to E2. Then E3 then catalyzes the transfer of your ubiquitin to the amino group of a lysyl residue of the target protein. So additional rounds of ubiquitination result in a lot of ubiquitination in the process. So this mechanism, okay, would be present at its amino terminal effects whether a protein is ubiquitinated. Okay. Your amino terminal, methine or serine, retards, whereas aspartate or arginine accelerates ubiquitination. So for ubiquitination, what would be, how do we call this? It facilitates, would be your aspartate or arginine, and what distracts would be your methyl or serine or your serine. Degradation occurs in a multi-catalytic complex of proteases known as the proteasome. Okay. So your glucose alanine cycle would be this one. Okay. So you have your glucose in the bloodstream. Then in the muscle, it gets converted to your pyruvate and eventually alanine as it receives your amino acids. This alanine goes out and goes to the liver as it becomes your, again, pyruvate, so it can be reutilized as your glucose. The end product, nitrogen, would then be converted to your non-toxic urea. And this hepatic gluconeogenesis from your alanine is the one significant, that's why it's one that is mentioned is higher than from all other amino acids. Okay. And its capacity for saturation is 20-30 times its physiologic level. Capacity of your liver for gluconeogenesis from alanine. Okay. As you will know what is alanine in clinical, this is what you will know as one of your liver function tests. So this is your ALT, as alanine concentration is measured. That's why it's important to measure it. Okay. Okay. Following a protein-rich meal, okay, does plant tissues release your amino acids? From the peripheral muscles, extract amino acids. So in both, they are predominantly branched chain amino acids. Okay. What do these branched chain amino acids do in nitrogen metabolism? So in the fasting state, they provide brain with an energy source, and in the fed state, they are extracted predominantly by your muscles. Okay. Digestion of dietary proteins. Okay. Proteins are hydrolyzed to amino acids for absorption to make them smaller. Proteolytic enzymes responsible for degrading proteins are in the stomach, pancreas, and small intestine. Okay, we will know later. Okay. In the stomach, okay, you know that they contain gastric juices, and their star enzyme would be your pepsin. The role of your HCl will kill your bacteria because of its acidity. Pepsin is from pepsinogen. Aside from killing bacteria, your HCl will activate your pepsin. In your pancreas, there are pancreatic proteases. They further cleave your polypeptidase initially digested by the stomach. Okay. Your enteropeptidase from the intestine would activate then your pancreatic enzymes such as your trypsinogen to its active form. Okay. Further, in the small intestine, you have your aminopeptidase cleaving further your oligopeptides to produce your free amino acids. See, these free amino acids and dipeptides are absorbed in your epithelium of the intestines. Okay. These are further hydrolyzed in the cytosol before being released to the portal system. Okay. So this one would be the fate of your amino acids. So your kidney excretes your nitrogen through serine, alanine to the liver, urea and glucose would be produced. Okay. First, the muscle, glutamine, goes to your intestines, also produce your alanine. Your muscles, glucose-alanine pathway will go to the liver. Muscle produces valine, goes to the brain. So these are your pathways of your different amino acids as it becomes your nitrogen, as it's converted to nitrogen. Okay. In lower life forms, okay, they also need to excrete nitrogen. And we have different ways to excrete nitrogen. Okay. For the fish, you have your ammoniteric. So they excrete ammonia. So they are compelled to excrete water continuously so that they would facilitate excretion of the highly toxic ammonia. Okay, that is for fish. So they are ammoniteric, meaning they excrete pneumonia. Okay. For birds, so since they cannot throw away water and conserve it, they maintain low weight and excrete uric acid as your semi-solid guano. So birds are uricotelic. Okay. Our species and other land animals are ureotolic. And this means we excrete non-toxic, water-soluble urea. Okay. And urea, as you will eventually know in your further studies in your medical school, urea would signify renal disease as a consequence of impaired renal function. If you cannot excrete, if the kidneys cannot excrete your nitrogen and convert it to your non-toxic urea, okay, or urea, it's accumulated because it cannot be excreted, it is just produced, then it will cause disease. Okay. Once urea is produced, it can occur is produced in 4 stages. Okay. The first is transamination. Okay. The second is oxidative deamination of glutamate. Third is through ammonia transport. And 4 would be reactions of the urea cycle. Okay. Okay, let's discuss them one by one. So what's transamination? So transamination interconverts pairs of alpha amino acids and alpha keto acids. Okay. All participate in transamination, all amino acids, except for 4. Lysine, threonine, proline, and hydroxyproline. Okay? So, all are transaminated except the 4. Okay. Reversible reaction, and these also function as your amino acid biosynthesis. Okay. So, transfer, again, what is transamination? It interconverts pairs of your alpha amino acids and alpha keto acids. So, for the nitrogen gets transferred into the other as your proteins are transferred. So transamination. Okay. You have your coenzyme pyridoxal phosphate. Okay, what does it do? So of course, your coenzyme is a catalyst. So it's present at the catalytic site of aminotransferase and of many other enzymes that act on your amino acids. Okay, this bound PLP serves as a carrier of your amino groups. Okay. Following the removal of your alpha amino nitrogen by transamination, the remaining carbon skeleton is degraded. Okay. Okay. So, for example, your alanine pyruvate aminotransferase and glutamate alpha ketoglutarate aminotransferase, these 2 enzymes would catalyze the transfer of amino groups to pyruvate, thereby forming alanine, to alpha ketoglutarate, forming glutamate. So, pyruvate and alanine would become alanine as it transfers. Your alpha ketoglutarate becomes glutamate as it undergoes aminotransferase from your amino to your keto acid. Each aminotransferase is specific for one pair of substrates, okay, but not non-specific for the other pair. Okay, wait. Okay, so since alanine is also a substrate for glutamate, all amino nitrogen from amino acids that undergo transamination can be also be concentrated in glutamate. Okay. Again, this is important because L-glutamate is the only amino acid that undergoes oxidative deamination at an appreciable rate that is significant in mammalian tissues. That's why your glutamate is an important amino acid. The formation of ammonia from your alpha amino groups thus occurs mainly via the amino nitrogen of your L-glutamate. Okay, so this would be the main source of your formation of your ammonia. Hold on, wait. I'll just answer a call. Okay, so L-glutamate dehydrogenase is also is central, therefore, in nitrogen metabolism. Okay, this is the transfer of amino nitrogen to alpha ketoglutarate, which forms your L-glutamate. Okay. As ammonia is catalyzed by your L-glutamate dehydrogenase, which can use either NADP plus or NAD plus. Okay. This conversion of your alpha amino nitrogen to ammonia by the concerted action of glutamate aminotransferase and your dehydrogenase is often termed as transdeamination. Okay. Its activity is inhibited by ATP, GDP, and NADH, and activated by ADP. Okay. Reversible reaction and also functions in amino acid biosynthesis. Okay. These would be the fates of your amino group. Okay, so your glutamate would be acted on by your glutamate dehydrogenase and will be transdeaminated to alpha ketoglutarate. Okay, same with your alanine and alpha ketoglutarate would become pyruvate and glutamate, and so on and so forth. Okay. Amino oxidases remove nitrogen as ammonia. Okay. L-amino acid oxidases of your liver and kidney convert amino acids to an alpha amino acid that decomposes to an alpha keto acid with release of your ammonium ion. Okay. This reduced flavin is reoxidized by molecular oxygen, forming your hydrogen peroxide, which then is split to oxygen and water by catalyst. Okay, so it will start with an amino acid, then it will be acted on by your oxidase and produce an amino acid. It will release an ammonium ion. Okay. And then this flavin would be reoxidized by oxygen, which forms your hydrogen peroxide, which is split by oxygen and water by a catalyst. Okay. What if we have ammonia accumulating in our body? It's life-threatening. Okay. Because we could only tolerate up to 10 to 20 grams per deciliter in blood. Okay. Toxic to our central nervous system. So it cannot be absorbed into our blood and tissues and should be rapidly removed and be converted to urea ideally. Okay. Should the portal blood bypass the liver, systemic blood ammonia may rise to toxic levels. So this severely impairs your liver function. Okay. Some symptoms of ammonia intoxication. Okay, you know that it affects your CNS. So it would produce tremor, it would produce slurred speech, affect your vision, affect your sensorium. Of course, ultimately, it could produce death. Okay. Since it's toxic to the brain because of your alpha ketoglutarate forming glutamate. As we have mentioned, glutamate is very important in mammalian biosynthesis of your amino acids. The resulting depleted levels of your alpha ketoglutarate impairs your function of your TCA cycle in your neurons. Okay. Your alpha ketoglutarate acts on your carboxylic acid cycle in neurons. Okay. Glutamine synthase fixes ammonia as glutamine. So how do you get out of it? So formation of glutamine is catalyzed by your glutamine synthase. Okay. Since your amide bond synthesis is coupled to the hydrolysis of ATP to ADP, the reaction strongly favors glutamine synthesis. Okay. And one important function of your glutamine is to sequester ammonia in a non-toxic form. Okay. Don't interchange. Glutamate is the toxic one. Glutamate forms alpha ketoglutarate, forms ammonia. Your glutamine sequesters ammonia. Okay? Then we add your other amino acids. Okay. As your hydrolytic release of the amide nitrogen of glutamine as ammonia is catalyzed by glutaminase, is strongly favored by glutamate formation. So this action of glutamine synthase and glutaminase catalyzes the interconversion of free ammonium ion and glutamine. Okay. And one similar reaction would be the catalyzation of your L-asparaginase. Okay. As shown in this diagram. Okay. So, how then can we maintain your acid-base balance as ammonia is formed and secreted? So, excretion into urine of ammonia produced by renal tubular cells facilitates cation conservation and regulation of acid-base balance. Okay, you know that ammonia production from your intracellular renal amino acids, especially your glutamine, increases in metabolic acidosis and is decreased in metabolic alkalosis. And you have learned extensively in physiology what metabolic acidosis and metabolic alkalosis is. Okay. And one, of course, in the excretion of urea, and we will look at the urea cycle. Okay. And your urea cycle is a series of reactions that will produce urea from ammonia, from a toxic ammonia to the non-toxic urea form. Okay. So this cycle was the first metabolic cycle that's discovered. In mammals, it only takes place in the liver. Okay. Hence, the importance of your liver function test, as I have mentioned, your AST and your ALT measurements. Okay. And what's the function of your urea cycle? Okay. They cannot, of course, you have known that ammonia cannot be removed easily, and you have to be converted to other substance. For mammals, it's urea. Okay. If, of course, your urea cycle is not effective or it's deficient, it would become toxic. It may be inborn error, genetic disorders of the liver, or acquired failure of the liver, as we will see later. Okay. And this liver failure, okay, of accumulated nitrogen, of course, you know, hyperammonemia would affect your CNS. So it will lead to your hepatic encephalopathy or neuropsychiatric abnormalities. Okay. So that's the biochemical basis of your NSEF in this case. Okay. And what are the reactions? Okay. Know these 5 reactions. The urea cycle would consist of 5 reactions. 2 would be found in the mitochondria. 3 would be cytosolic. Okay. And in the end, these 5 reactions would do the following. Okay. It converts your 2 amino groups, one from ammonia and one from aspartate, and a carbon atom from your bicarbonate to relatively your urea, causing 4 high energy phosphate bonds. 3 ATP hydrolyzed to 2 adenosine phosphate and 1 AMP. And the main carrier of these carbon nitrogen atoms would be your ornithine. Okay. Okay. Urea as the major end product of nitrogen catabolism in humans. Okay. So, very energy requiring reaction. Your 3 moles of NATP would only produce 1 mole of urea. Plus, 1 mole each of your ammonium ion and an alpha amino nitrogen. Okay. What functions as your enzyme activator? It's your N-acetyl-glutamate. Okay. What if it's energy expend? You see how important it is. The body looks at your urea production, right? If you have, think of your ATP as money to spend. So even if it is an expensive process or it's an expensive thing to produce your urea, your body still sees it as a need. Okay, even if it costs the body 3 ATPs. That's how important the urea cycle is. Okay. What's the initiator? And usually the initiating step is a rate limiting step. Okay. Your carbamoyl phosphate synthase 1 would initiate your urea biosynthesis. Okay, that's condensation of your carbon dioxide, ammonia, and ATP to form your carbamoyl phosphate, catalyzed by mitochondrial carbamoyl phosphate synthase 1. Okay. It has to have N-acetylglutamate for it to become active. Okay. Only in the presence of N-acetylglutamate, which makes it more affinity, increase its affinity to the synthase for ATP. Okay. Initial step would already require 2 moles of ATP. Okay. Rate limiting. What's rate limiting? It has to have 2 moles of ATP. It has to occur in the presence of your acetylglutamate. It has to occur in your mitochondria. Otherwise, no urea cycle initiation. Okay, so where are we? So in the mitochondria, carbamoyl phosphate will be reacted on your carbamoyl synthase 1. In the liver mitochondria, N-acetylglutamate should be present with 2 ATP. Reaction 1. Rate limiting step of your urea cycle. Okay. What's carbamoyl phosphate synthase 1? Any defect, of course, in your enzyme carbamoyl phosphate synthase would be responsible for a relatively rare metabolic disease, hyperammonemia type 1. If you have rate limiting step and there's no urea cycle, what do you think will happen? Of course, your ammonia will accumulate. Okay? So, a defect in carbamoyl phosphate synthase 1, rare, would produce your hyperammonemia type 1. Okay. Then, your L-ornithine transcarbamylase would transfer group of your carbamoyl phosphate to ornithine, forming citrulline and orthophosphate. This reaction too still occurs in your mitochondrial matrix. Okay, your ornithine transcarbamylase transfers your carbamoyl group of carbamoyl phosphate, and what will happen? Citrulline and orthophosphate. Okay, where is this? Okay, here's your ornithine And it would be your ornithine transcarbamylase reaction 2. As it goes outside the mitochondria, what will it produce? It will produce your citrulline and what's the other one? Citrulline and orthophosphate. Okay, we're outside the mitochondria for after our second reaction. Okay. Okay. So, what's the role of ornithine? Okay. Your hyperornithinemia, hyperammonemia, and homocitrullinuria syndrome. That's your triple H syndrome. Results from the mutation of your ornithine 1 gene, which encodes the mitochondrial membrane ornithine transporter. Okay. What happens? Failure to import your cytosolic ornithine into the mitochondrial matrix renders the urea cycle inoperable. So if your transporter does not work, cannot provide an interface between your cytosol and your mitochondria, your reactions cannot proceed. Thereby, your urea cycle will also not proceed. Okay, so that's the function of your ornithine transporter. And if that has a problem, you have your HHH. You would have an excess of your ornithine, you have an excess of your ammonia, and you have your excess of your citrulline. Where? In your cytosol. In your mitochondria. Okay. How about ornithine transcarbamylase as a deficiency? This X chromosome linked deficiency would then lead to hyperammonemia type 2. So this is a defect in your ornithine transcarbamylase. Okay. So what will happen? So they will exhibit symptoms of hyperammonemia and would be averse to high protein foods. Okay, why? Okay, levels of glutamine are elevated in blood. Again, what's glutamine? It's the one that responds to elevated tissues of ammonia. Elevated in blood, elevated in CSF and urine. Okay. As a result of enhanced glutamine synthesis. Okay. Next would be your citrulline plus aspartate forms arginosuccinate. Okay, this links aspartate and citrulline via amino group of aspartate and provides the second nitrogen of urea. So this is another reaction that will require ATP. Okay, and subsequent displacement by aspartate will form citrulline. Okay. Then you have your cleavage of argininosuccinate catalyzed by your argininosuccinase, proceeds with retention of nitrogen in arginine and release of the aspartate skeleton as fumarate. Then fumarate is recycled back to aspartate. So addition of water to fumarate forms your L-malate and subsequent NAD plus dependent oxidation of malate forms oxaloacetate. Okay, these two reactions are analogous to reactions of your citric acid cycle and are catalyzed by cytosolic fumarase and malate dehydrogenase. Okay, then your oxaloacetate by glutamate aminotransferase then reforms aspartate. Okay, it goes back. Where are we? Here is your argininosuccinate. Argininosuccinase produces fumarate. goes to your citric acid cycle. Then you have your arginine. Then it goes back to your arginase. Okay. Your arginine is produced from your argininosuccinate. Okay. Your ASL gene is located on chromosome 7. Okay. If deficient, you have your elevated levels of argininosuccinate in blood, in the cerebrospinal fluid, and urine. Okay. You have your arginosuccinic aciduria associated with friable tufted hair. So this is your Trichorexis nodosa. See, cleavage, arginine releases urea, okay, and reforms ornithine. So hydrolytic cleavage of the guanidino group of arginine catalyzed by liver arginase releases urea. And the other product you're familiar with is ornithine. Re-enters liver mitochondria and again, part of your urea synthesis. Okay? So that is the urea cycle. Starting with your carbamyl phosphate 1, acted on by an N-acetylglutamate to ATP. Ornithine transmacromolase acts on your ornithine to produce your citrulline and your other phosphate groups. Then your arginosuccinate synthase acts on your aspartate to produce your arginosuccinate. Arginosuccinase becomes arginine. Your arginine eventually produces your urea and your orthonine, and again the urea cycle. Okay. So ornithine and lysine are potent inhibitors of arginase and competitive with arginine. And this also serves as the precursor of the potent muscle relaxant nitric oxide in a calcium-dependent reaction catalyzed by nitrogen synthase. Okay, so what's arginase? Hyperarginemia is an autosomal recessive defect in a gene for arginase. Okay. Hyperarginemia symptoms do not appear until age 2 to 4 years. Okay. Blood and cerebrospinal fluid levels of arginine are elevated. Okay. As I've mentioned, rate-limiting step of the urea cycle is your carbamyl phosphate synthase 1. Okay, and this is determined by your coenzyme N-acetylglutamate. Okay, N-acetylglutamate level is dictated by the rate of synthesis from your acetyl-CoA and glutamate and its rate of hydrolysis to acetate and glutamate. So these reactions are catalyzed by N-acetylglutamate synthase and N-acetylglutamate hydrolase. Okay, what's N-acetylglutamate synthase? N-acetylglutamate is essential for carbamyl phosphate synthase 1 activity, as we have mentioned, as a coenzyme. Say, the next gene encodes your N-acetylglutamate synthase, which catalyzes the condensation of acetyl-CoA with glutamate. Okay, any defect would result, as we have mentioned in the rate-limiting step, severe hyperammonemia, which may respond to administered N-acetylglutamate. So here, you administer your N-acetylglutamate and your deficit is gone. It will proceed. Okay. As I mentioned, carbamyl phosphate, rate limiting step, if you provide the coenzyme, you can proceed with the urea cycle. Changes in diet can increase concentrations of individual urea cycle. Okay, of course, starvation elevates enzyme levels to cope with the increased production of ammonia. Okay. Okay, what are the other metabolic disorders? So all defects in urea synthesis will result in ammonia intoxication. Okay. The effects are most severe when the metabolic block occurs at the first two reactions. For if citrulline can be synthesized, some ammonia has already been removed by being covalently linked to an organic metabolite. Okay. So it's reactions 1 and 2, that's carbamyl phosphate 1, 2 carbamyl phosphate synthase 1 to ornithine, and your ornithine to produce citrulline. Those are the critical reactions. Okay. So clinical symptoms common to all urea cycle disorders would include the following. Okay. Vomiting, aversion to high protein foods, intermittent ataxia, irritability, lethargy, and severe mental retardation. Okay. Of course, improvement would be accompanied by low protein diet or small frequent meals to avoid the sudden increase in blood ammonia levels. Okay. What else? They are fed high doses of benzoic acid and phenylacetic acid, okay, which are excreted in the urine as conjugation products with glycine and glutamine, respectively. What else is promising for defects in urea biosynthesis? Gene therapy, okay, to rectify defects in enzymes of the urea cycle is promising. Okay, some have been obtained, for example, in animal models using an adenoviral vector treating citrullinemia. Okay, so for those who prefer table visualization of the urea cycle, these are the 5 reactions: reaction, product, catalyzed by, and where it occurs. Okay? So this is just a summary of the urea cycle in tabular form. Okay? And this one would be your whole catabolism of your amino acids as we have discussed it this afternoon. Okay, so we have discussed for this lecture your fate of amino acids catabolized in our body, the enzyme systems, ubiquitin, transaminase, glutamate dehydrogenase. It's the role of the stomach, pancreas, and small intestine in how we degrade the proteins, the urea cycle, sources of nitrogen atoms, and symptoms of hyperammonemia. Okay? Any questions for amino acid catabolism? Any questions for amino acid catabolism? None. Do you want a break for carbon skeletons or you want to proceed?
Proceed, doctor.
Proceed, doctor.
Okay. So, in relation to our previous lecture, we'll have your carbon skeletons of amino acids. Faith. Okay. As you have learned your protein from the diet, amino acids in relation to nabuksan din. Okay. Your amino acids would produce your urea. As it becomes synthesized, some would be utilized by the body through your proteins, and the rest would become carbon skeletons and further converted into your glycogen or fat. And further provide our energy needs. Okay? That's the role of your amino acid. Okay. For this lecture, we will look at your amino acid catabolism. Okay. Remember, your removal of alpha amino nitrogen by transamination, remember these 4 exceptions in the previous lecture, is always the first catabolic reaction, except for the following: proline, hydroxyproline, threonine, and lysine. Okay. Okay. Catabolism of the carbon skeletons of amino acids are done so that their precursors can be metabolized to carbon dioxide and water, or to be used in gluconeogenesis. So this accounts for 10 to 15% of metabolic energy that's generated by animals. Okay. So there are different classifications of your amino acids. So it could be glucogenic, it could be ketogenic, or carbon skeletons are degraded to pyruvate, alpha-ketoglutarate, succinyl-CoA, fumarate, or oxaloacetate. And they are glucogenic amino acids because they are glucose precursors. On the other hand, your ketogenic amino acids are carbon skeletons broken down to acetyl-CoA or acetoacetate and can thus be converted to fatty acids or ketone bodies, thereby ketogenic amino acids. Okay. And this would be how your amino acids are produced and utilized along the citric acid cycle. Okay, so along the citric acid can be used, can enter the citric acid cycle as either your precursors, as either your enzymes, or your as your substrates, so that your citric acid cycle could proceed. Okay. So you have your glucogenic and ketogenic and glucogenic and ketogenic. As you can see in the table, the only solely ketogenic amino acid would be your leucine. Okay. Others would be glucogenic and ketogenic. Others would be purely glucogenic. Okay. So these are your amino acids that are converted to pyruvate. Okay, alanine, cysteine, glycine, serine, threonine, and tryptophan. Okay, amino acids converted to oxaloacetate would be your aspartate and your asparagine. Converted to alpha ketoglutarate would be your arginine, glutamate, glutamine, histidine, and proline. To fumarate, aspartate, tyrosine, phenylalanine. To succinyl-CoA, isoleucine, methionine, and valine. To both acetyl-CoA and or acetoacetate, isoleucine, leucine, threonine, lysine, phenylalanine, tryptophan, and tyrosine. Okay, and this would be your where it comes in. Okay. So how is it converted to pyruvate? So transamination forms pyruvate, which can then be decarboxylated to acetyl-CoA. Okay. Your alpha ketoglutarate group or your oxaloacetate undergoes transamination from alanine to pyruvate, would produce your aspartate and eventually your acetyl-CoA. Okay. Your glycine splits to carbon dioxide and ammonia, and your N5-N10 methylene tetrahydrofolate. So your glycine would be acted on, produce your ammonium ion, and then your byproduct would be your N5-N10 methylene tetrahydrofolate. Okay. So this would be your transamination of glycine to glyoxylate with your glucose or your alanine. Okay. Serine. So this is your serine. It would be transferred, it would become pyruvate by the enzyme serine hydratase. Okay. Degraded to glycine and your N5-N10 methylene tetrahydrofolate. Okay. Cysteine converted to cysteine, and your cysteine is converted to via two pathways, either direct oxidative pathway or your transamination pathway. Okay. Cysteine would be converted to cysteine by your cysteine reductase. It will be reduced by your NAD plus and your NADH and would produce your cysteine. Okay. Your cysteine would have two pathways, either a direct oxidative pathway or a transamination pathway. Okay. So direct oxidative pathway would be from cysteine acted on by cysteine dioxygenase, thereby cysteine sulfonate acted on by a transaminase, and then produce your pyruvate. For transamination pathway, you have your cysteine by a transaminase would become your pyruvate and produce your thyropyruvate. Threonine is converted to your acetyl-CoA through acetaldehyde oxidation to acetate, converted to acetyl-CoA. Okay, so your threonine gets acted on by threonine aldolase, would have your acetaldehyde, and this acetaldehyde would be converted to acetate by aldehyde dehydrogenase and would have your acetate thiokinase and eventually have your acetyl-CoA. Okay, so this is your threonine, glycine. Okay, would convert to serine, and acetaldehydes, acetyl-CoA, and all will end up to be pyruvate. Okay. Your oxaloacetate pathway. Aspartate transamination forms oxaloacetate, and your asparagine undergoes hydrolysis to aspartate, and your aspartate again forms your oxaloacetate. Okay, so aspartate and asparagine. So your asparagine becomes your asparaginase as aspartate by your asparaginase. Okay. Once it becomes aspartate, it undergoes a transaminase reaction and produces your oxaloacetate. Okay. So, amino acids converted to alpha-ketoglutarate. Okay. Your glutamate transamination forms alpha-ketoglutarate. Your glutamine undergoes hydrolysis to glutamate. Okay. So, for it to be your alpha-ketoglutarate, your glutamine will be acted on by your glutaminase to produce your glutamate. Once it becomes your glutamate, you have your transaminase acting on your glutamate, and it will produce your alpha-ketoglutarate. Okay, proline. This is oxidized to dehydroxyproline, which adds forming glutamate gamma semialdehyde. This is then oxidized to glutamate and transaminated to alpha-ketoglutarate. Okay. So we start with proline, acted on by your proline dehydrogenase, then will produce your glutamate semialdehyde. Okay, then this would be transaminated, oxidized to glutamate, and then your glutamate will undergo transamination to your alpha-ketoglutarate. You will see that it also goes back to glutamate before it becomes your alpha-ketoglutarate. Arginine, converted to ornithine, which then undergoes transamination to glutamate semialdehyde. So ornithine undergoes transamination. It will become your glutamate semialdehyde. And what will happen? Your glutamate semialdehyde will become, if you will follow the previous, will become what? It's also here. Will become glutamate and then become your alpha-ketoglutarate. Okay. Histidine. It's a non-oxidatively deaminated, then hydrated, and its imidazole ring cleaved to form your N-formimino-glutamate. Formimino group is then transferred to your TH4 to form your N-formimino TH4 and glutamate. Okay, so this one's different. Histidine undergoes uroconate by histidase, then uroconase for imidazole 5-propionate. Okay, then N-formimino-glutamate by your imidazoline propionate hydrolase before it becomes your glutamate, before it becomes your alpha-ketoglutarate through your glutamate formimino transferase. Okay, and excretion of your fig-glue following a dose of histidine can be used to detect folic acid deficiency. Okay, so here would be the pathway. Okay, glutamine, histidine would have your fig-glue, then your glutamate. Proline converted to glutamate semialdehyde and glutamate. Arginine produces ornithine, becomes glutamate semialdehyde, becomes glutamate. Glutamine becomes glutamate, then ketoglutarate, and then glutamate itself becomes alpha ketoglutarate. Okay? So those are intersecting and overlapping reactions. How about fumarate? So phenylaniline, first reaction in its degradation is its hydroxylation to tyrosine. So phenylaniline is hydroxylated to become tyrosine. Okay. What is tyrosine then? Transaminated to p-hydroxyphenylpyruvate, followed by concerted ring hydroxylation and side chain migration to form homogentisate with ascorbate as reductant. Aromatic ring opens and is hydrolyzed to fumarate and acetoacetate. Okay. Then what is the fate of tyrosine? Yeah, tyrosine would then be converted to fumarate through a lot of reactions. Tyrosine becomes p-hydroxyphenylpyruvate, homogentisate, malol acetoacetate, maliacetoacetate, fumaryl acetoacetate, fumarate, and then acetyl-CoA and acetate. Okay. So aspartate, we have learned your aspartate, also part of your urea cycle. As it goes into your cycle, it would be utilized, okay, through your arginosuccinate. Okay, phenylaniline converted to tyrosine, homogentisate, becomes your fumarate. Okay, you aspartate, as you have seen in the urea cycle, aspartate reacts with your arginosuccinate to become your fumarate. Okay, amino acids converted to your succinyl-CoA. Okay, methionine condenses with your ATP to form your S-adenosylmethionine. So SAM is an important methyl donor. Okay, L-methionine with your ATP produces your SAM. So methionine would have your removal of your methyl group forms. So with your S-adenosyl homocysteine, which is hydrolyzed to adenosine and homocysteine, homocysteine combines to serine to yield cystathione, which subsequently forms cysteine and alpha-ketobutyrate. Alpha-ketobutyrate is degraded to propionyl-CoA and then to succinyl-CoA. Okay, so your methionine produces your SAM, becomes your L-homocysteine, then cystathionine, then becomes your alpha-ketobutyrate, and then succinyl-CoA. Okay, so isoleucine is converted to propionyl-CoA, valine is converted to methyl-malonyl-CoA. Okay, mechanisms to be discussed with other branched chain amino acids. So your methionine would become alpha-ketobutyrate, isoleucine would be your propionyl-CoA, valine would be your methyl-malonyl-CoA. You will see later that all of that would be in the pathway producing your succinyl-CoA. Amino acids degraded to acetyl-CoA and or acetoacetate. Okay, lysine. So this has several pathways for degradation, but the pathway that proceeds via formation of saccharopin predominates in mammalian liver. Okay. The pathway involves transamination, oxidative carboxylation, and reactions similar to fatty acyl-CoA oxidation. Okay. So what's the most, this is the saccharopin route, lysine, becomes your saccharophil, becomes your L-alpha aminoadipate gamma semialdehyde, L-alpha aminodipate, alpha ketodipate, glutaryl-CoA, then your succinyl-CoA. Okay? Tryptophan. Okay. Carbon atoms of side chain and aromatic ring completely degraded by your kynurenine antralinate pathway. So initial reaction involves cleavage of indole ring with incorporation of 2 atoms of molecular oxygen by tryptophan oxygenase. So this is the fate of your tryptophan. Tryptophan N-formylkynurenine, L-kynurenine, 3-hydroxykynurenine, 3-hydroxyantranilate, 2-acrolyl 3-aminofumarate, oxaloactronate, and then ketoadipate, then acetoacetate. Tryptophan oxygenase, an iron 4-phenyl metalloprotein inducible in the liver by adrenal corticosteroid and by tryptophan. Feedback is inhibited by nicotinic acid with derivatives including your NADPH. Leucine, as we have mentioned, it's purely ketogenic amino acid. So this is degraded to HMG-CoA, which is converted to acetoacetate and acetyl-CoA. Okay, we will discuss this later with your other branch chain amino acids. Okay, branch chain amino acids. Okay, you have your valine, isoleucine, and leucine. Shares the first 3 reactions that employs common enzymes. Resulting products are then catabolized by distinct pathways. Okay, what are the first 3 reactions that are shared by your 3-branch chain amino acids? Transamination to corresponding alpha keto acids, oxidative decarboxylation to corresponding acyl-CoA, dehydrogenation by FAD to form a double bond. Okay, so these are your branch chain amino acids, leucine, valine, and isoleucine. Okay, leucine first. It becomes your alpha keto isocaproate, then isovaryl-CoA. L-valine becomes your alpha keto isovalerate, then isobutyryl-CoA. Isoleucine becomes your alpha keto B-methylvalerate, becomes your alpha methylbutyryl-CoA. Okay. Then all of that will become acetoacetate, acetyl-CoA. Okay. And this would be its fate in the citric acid cycle. Okay. What's the relevance? Okay. So amino acidopathies are almost associated with amino acid catabolism over synthesis. So what is always problematic would be how it is used rather than how it is produced. Okay. Amino acids present in the diet, especially if it's well balanced. Okay. But the utilization will result in accumulation of the amino acid and its metabolites. And of course, any accumulation would end up in toxicity. Okay? Okay, what are the common amino acidopathies that you have to know? So first would be your alkaptonuria. Okay, alkaptonuria would be the defect in tyrosine catabolism. Okay. What's deficient? Your homogentisate oxidase. And most striking manifestation is darkening urine that stands in the air due to the presence of homogentisate. So later in life, these people develop arthritis and connective tissue pigmentation. Okay. Next, acidopathy would be your phenylketonuria. Cool. This would be inability to convert phenylalanine to tyrosine. Okay, so if your phenylalanine is not converted to tyrosine, your phenylalanine hydroxylase would accumulate. This is primarily because of a defect in your enzyme phenylalanine hydroxylase. So this is your classic phenylketonuria. A major consequence is your mental retardation. A treatment, your diet should be low in phenylalanine. You know that your phenylalanine should be converted to tyrosine. Of course, if there's a break there, your phenylalanine will be accumulated. Okay. There are some alternative pathways, okay, of course, but still it would be limited. Heart disease. So defect in the intestinal and renal transport of neutral amino acids, including tryptophan. Okay, this manifests with pellagra-like signs and symptoms because of limited conversion of tryptophan to niacin. MSUD or your maple syrup urine disease. Defect is absence of branched chain alpha ketoacid dehydrogenase complex. And odor resembles maple syrup or burnt sugar. That's why the name. Brain damage develops unless properly treated with diet low in branched chain amino acids. Okay. And that would be the end of today's lecture for the two. Any questions for this afternoon?